everyone. Welcome to our channel Biology for All. In this video, I will be showing how to draw the stages of mitosis and the key features for each one of them. The first phase in mitosis is prophase. This is the longest phase and has quite distinct features. The cell has to be drawn round or oval in shape, slightly oval in shape. The nuclear membrane is drawn as a broken circle as the nuclear membrane disintegrates. Chromosomes are to be drawn thick and short as they condense. Also, it is necessary to draw chromosomes in even numbers. 4 is a good number. The other feature is a centrosome that starts moving towards the poles. So there are two centrosomes with centrioles creating spindle fibers between them. So once we finish all these features, then we can label the diagram as I have done here. Metaphase is the second phase. Here, the cell again has to be drawn round or slightly oval in shape as we have done in the prophase stage. The nuclear membrane has completely disappeared, so we are not drawing that here. Now we draw a dotted line in the center of the cell. Next, the centrosomes have reached the pole, so they are to be drawn at the poles and they have spindle fibers extending to the middle line. Next, we draw the chromosomes lined on the dotted line with the centromere on the line and the arms facing towards the poles. We can then extend the spindle fibers to the centromere of the chromosome. So once these features are completed, then the labeling can be done as shown here. Anaphase is the third and considered to be the shortest phase. Now here, the shape of the cell changes. We had drawn a round or a slightly oval shaped cell in prophase and metaphase. But here, the cell now elongates a bit. So we have to show an elongated cell here without again any nuclear membrane. The centrosomes are on the poles, and now the centromere of the chromosomes have split to separate the chromatids. So these chromatids are now being pulled towards the pole. The chromatids are now called as daughter chromosomes, and they have to be drawn in a V or L shaped. Now, most students make the mistake of drawing an inverted V in this way. This is not correct. So you have to make sure that the centromere is on the spindle fiber and the arms are being pulled. So it has to be in the arms have to be facing the middle portion of the cell. So once the diagram is completed with the features as mentioned here, then we again label the structures. The last phase is telophase. Here, the middle of the elongated part of the cell shows a small depression on both the sides. Since this is the last stage of karyokinesis, we have to show two nuclei that are forming here. So we show the broken circle as the nucleus, but we are showing two on each side of the depression. The chromosomes have to be drawn inside that, but now they are the unreplicated chromosomes, and the centrosome remains near the nucleus. There are no spindle fibers, and after completing this features of telophase, now it can be labeled entirely. The same features are to be drawn for a plant cell also, except that you have to add cell wall and you don't have to draw the centrosomes. The division in plant cells is referred to as anastral. as they do not have the esters as shown here, the diagram of metaphase in plant cell.